Great, great. It's nice to see so many smiling faces here in the room. It is my great honor to welcome you all to the first ever FDA MHRA Good Clinical Practice Workshop. I'd like to start the day off by providing three messages of thanks. First, to our colleagues in the CDER Small Business and Industry Assistance Group for all of their tireless efforts to make this engagement possible. Thank you all to SBIA. Second, to our colleagues from MHRA, the Medicines and Healthcare Products Regulatory Agency. Thank you for partnering with us to make this engagement the best that it could be. And thank you also for coming all the way over here. I know that if your travel system is anything like ours, that is nothing short of miraculous. And lastly, I'd like to thank all of our attendees, both here in person and online. The response to this event has been truly phenomenal, and it is my great honor to welcome you to this first ever collaborative event. My greatest hopes are that you find this engagement to be valuable, that you find the information that we share with you here today to be important, critical to improve your work moving forward, and that you see value in this collaboration between FDA and MHRA to be able to share ideas and move the concept of data integrity forward in the medical product development industry. Thank you once more for attending. I think we have a fantastic show for you today. I think you're going to really enjoy it. So our topic, as I'm sure you are all very well aware, is data integrity in global clinical trials. Are we there yet? Are we there yet is a yes-no question. That's kind of curious, isn't it? Yes, no, is it 50-50? Really, not even close. It's a question of grades. It's a dynamic. The answer really is maybe. Maybe with a dash of uncertainty, maybe with a side dish of ambiguity. That's why we're here today. It's such a compelling topic. It registered with over 3,000 attendees in the global environment. Absolutely incredible. So, why would over 3,000 people from 68 countries come together to hear about data integrity from MHRA and FDA? Why such an overwhelmingly positive response to this question? It's not like we're going to share some shining beacon of hope that's going to just sunshine and rainbows for the entire industry. There's no secret. This is a conversation, moving the ball forward just one step at a time. So what is the promise of today? What opportunities are there for us in this room and online today, just this minute, to use this conversation to make tomorrow a better day? Clinical research is a global industry, more so now than ever before in our entire history. We're all partners in public health, as regulators, as sponsors, as applicants, as CROs, as clinical investigators, as patients, partners in public health. And today, technology, digital health, electronic systems, just the changing dynamic of the global industry, it's changing the business that we work in front of our very eyes. This big world is becoming so much smaller, and it's becoming smaller at a rate that is frankly a little scary. So the promise of today is just taking that one next step in data integrity. That's a big idea. So big, in fact, that it was able to bring together FDA and MHRA for this first of its kind engagement. The idea there is just so compelling. But we still haven't gotten to what data integrity really means. What does it really mean in this context? How are we going to view data integrity for this conversation here today? Data integrity, it's not black and white. It's not yes or no. It's an incredible continuum. There are shades of gray. And the shades of gray here are really what drive the conversation. Those nuances, they become very sticky shades of gray that are just thought-provoking and compelling. That uncertainty is incredible. So data integrity for us today really just means reliability, interpretability. It means traceability of data and the principles of Alcoa. And why do we care? Because if we can move the needle on data integrity just one little bit, we can increase the certainty in drug development. A really compelling idea. The promise of today is that when we can take that concept of data integrity as a global industry forward one step, we can increase the clarity that we have, using that clarity to drive confidence in our drug development programs, and using that confidence in the way that we're moving forward as an industry to foster continuous improvements, to drive further quality moving forward. Clarity, confidence, and action, so much more rewarding than ambiguity and uncertainty in inaction. So make no mistake that these are challenging issues. It's a little scary. It's ever-shifting, ever-changing environment. Data integrity is something that can never really be completed. There's no finish line. That's why this is a conversation that we need to continue to have over the course of time. Even you, as an applicant, when you have the approval for a new drug product, does that really mean that you have reached that nirvana state of data integrity? Not really. Maybe, but not really. It's a conversation, a continuum over time. My request is that as you participate in today's engagement, stay open-minded. Open-minded to the ideas you're hearing, open-minded to how you might change your practice moving forward, open-minded to how this relationship might be able to continue a conversation in the future for further change. And we, as regulators, will be open-minded in how we might be able to best protect the public health. So, are you ready to dive in, to wrap your arms around that uncertainty of data integrity? Those shades of gray right now, for the first time in this conversation, are things that we're going to start to paint in color. And that is my great hope for this conversation. So with that, today I say thank you for attending this first ever joint collaboration between FDA and MHRA on Good Clinical Practice Workshop. And I would like to pause and introduce our keynote speaker, Dr. Robert Temple. Dr. Temple is, there we go. Dr. Temple is a professional who really needs no introduction, but we'll get one anyway. He currently serves as the Deputy Director for Clinical Science at FDA's Center for Drug Evaluation and Research. He has a wealth of experience in clinical trial design and data integrity in the global domain. And he serves in a dual role, also as the Acting Deputy Director of the Office of Drug Evaluation, and is responsible for cardiorenal, neuropharmacologic, and psychopharmacologic drug products. Here today to address data quality and bias, please join me in welcoming Dr. Temple. Thank you.
Thank you. I'm going to talk from here because it's easier to see this, and apparently it's easier for me to be on that. Okay, can you hear me? Yep. Okay. I'm basically going to start with an anecdote, something that happened in 1980, 82, or thereabouts, that reminded us of what you need to know about how a study was conducted. It was a very instructive example, and we'll see what you think. It's perfectly obvious that the data we review, the clinical, the chemistry, the drug quality, pharm tox, has to be real and accurate to be informative. Otherwise, how would we? Otherwise, how would we learn anything from it? So it can't be made up. We need to know that there were real studies, the measurements were done and recorded. We don't like to think that critical things are omitted. And there are usually records that report all this, lab data, protocols, case reports. You can look at all of those. There have been cases, and we know about them, where some of these measures were fabricated. Sometimes that was easily detected. You saw the same electrocardiogram in consecutive weeks. That's a clue that somebody wasn't doing it. We've seen blood pressures that were all identical for a whole group of people in a trial. So we know about that. You also want to be sure all of the things that were supposed to be done were done. If one of the questions was about whether the drug makes you sleepy, you got to know that people asked about sleepiness. So we're also interested in those things. And then finally, and very important, we want to be pretty sure that there's no bias in the data collection or analysis. And nowadays, partly stimulated by this experience, people write out in advance how they're going to do it, who knows what, and all that sort of thing. And it's possible to check it to a degree, but it's worth remembering that if somebody gets information that you didn't know they were going to get, like who was on what, and they're doing something subjective, that can make a big difference. So I was going to go through some of the things. Obviously, you want to know that all the data has been collected right. The investigators will ask about the side effects, clinical endpoints, using protocol-specified queries or outcome scores. You want to know those things have all been done. And you want to know that they're performed optimally. You can easily screw up a 6-minute walk if you don't do it right, if you don't give the patient appropriate guidance. Usually that just makes noise and it messes up the study, makes it hard to show anything. But if you know what drug the person's on, it could introduce bias. And we always worry about those things. So we know that. You blind studies for a lot of very good reasons. You have control groups for good reasons. Many endpoints and adverse effects are subjective. You don't want the knowledge of the treatment group to affect either the finding or the strength of the finding or any of those things. You don't even want sort of innocent bias, an expectation on the part of the investigator that things are going to get better, even if they don't know the assignment. You want it to be completely objective. So we always want to know all the details of blinding to the treatment, about the firewalls that prevent sharing this knowledge. Sometimes some people have to be unblinded. If there's a terrible side effect or something, you've got to know whether the person was on the drug or on the treatment, but you don't want that information to be shared with anybody doing the other things. We usually minimize bias by blinding trials, but trials are designed to protect against bias analyses, but how to handle data, and how to handle data is usually specified very carefully in the protocol. For example, how, if at all, can a patient be removed from the study or the analysis? Is that even possible? Is there any basis for removing a patient? You've got to understand exactly how that's going to be done. You have to know how study dropouts will be dealt with. It has to be in the protocol and specified. You have to know who exactly knows the patient assignment. Somebody where they're giving out the drug does, but is that all hidden from everybody else? Who, if anybody besides the investigator, assesses endpoints? It's not uncommon, for example, to have a central group that takes the endpoint and makes a judgment about whether the endpoint was real or what was found. I'm going to show you an example of where there was a cause of death analysis. Who's doing that? And are they blinded? That's a secondary analysis. It follows the one made by the investigator. But if that's done centrally, who knew? Who knew what? And then, of course, we know this now, you have to have your planned study endpoints. They have to be ordered in a way. You can't just go picking and choosing and inventing new ones. It's all part of a statistical analysis plan, but sometimes things are changed, and you have to know the exact circumstances. And a lot of what's important and what can go wrong is illustrated by the experience I'm going to describe. And it occurred well before we really understood how a trial can be biased. I'm sorry to use the brand name. It was called the anturane reinfarction trial. Anturane is sulfenpyrazone, a drug that lowers uric acid. And the trial was done in the late 70s, and we evaluated it in the early 80s. It taught us all kinds of things that we really were naive about. It taught us about choosing the primary endpoint up front and not changing them after you look at the data. It talked about the risk of unplanned subsets, the timing of the analysis. It taught us about intent to treat and accounting for all patients in analysis. It taught us about blinding the results of interim evaluations and using them perhaps to alter subsequent analytic plans. Very treacherous, unless you reserve some alpha for it. It also taught us about the use of after-the-fact eligibility assessments. I'm sure nobody in the room even imagines that you can declare a patient ineligible after the study's over, but well, you see wait till you see what we did. And it taught us about the treachery of cause of death assessments, which we're very careful and nervous about. The angioedema reinfarction trial was, in many respects, one of the most carefully conceived and designed trials at the time. And it was one of the first large outcome trials that, as far as I know, was ever carried out by a drug company. We'd seen such trials before, but they were by NIH or the VA or foreign government agencies and things like that. It had several very distinguished review panels that helped design the trial and assess it as it was going, world-famous statisticians and things like that. But it nonetheless proved to be a model for how bias can be introduced into a trial, and we don't actually know exactly how it happened. A critical person who was responsible for the analysis died in an automobile accident at about the time we were looking into all this. I won't go into the nature of the suspicions that arose, but the problems were there were changes in the assessed study endpoints. There were late deletions of patients who had endpoints, who died, and the deletions all favored the treatment. And then there was cause-specific mortality, which was assessed by the investigator at the time of the event, but then was reevaluated, and we don't even know who did the reevaluation, but we do know it was terribly biased, as I'll show you. We don't always insist on an intent-to-treat analysis, but when events, even fatal outcomes, occur in patients on treatment or removed for some reason because of poor compliance or eligibility, you've got to worry about how that decision was made, whether it was planned in advance, and so on. Obviously, this is a particular problem, fatal problem, when the people deciding on exclusion aren't blind to the treatment, because you know exactly how to do it. So you need to know exactly who had access to unblinded data, who made all the decisions about patient inclusions, and all that stuff. And part of inspecting the study is knowing all that. I've got much more than we need to go through, but a couple things about the angioedema reinfarction trial. They first started thinking about it in 1975. Sulfenpyrazone, angioedema, was known to delay diminish platelet aggregation and do various other things. An odd feature was that as early as 1976, there was an inquiry to us about possibility of early stopping. Why did they ask that then? They didn't have any data yet. Anyway, by 1977, all of the 1,500 patients in the trial were entered, and they published their interim results with a showing a nearly 50% reduction in cardiac deaths, which was not corrected for multiplicity or early looks, and it was almost all due to a reduction in sudden death. So they already had an inkling of something that was going on. Anyway, they reobtained informed consent from everybody. They actually published this results and made them public, but nonetheless, they kept the trial going. All that was very odd. People wouldn't do it that way nowadays. Finally, in 78, they filed the NDA, an advisory. That was before the study was completed. At the interim look, our advisory committee said, no, not yet, can't do it. Then they finished the trial, resubmitted the NDA, and now the Cardiorenal Advisory Committee recommended approval for prevention of sudden death in the first 6 months. They didn't agonize about how that was an after-the-fact subgroup. And that was clearly not the planned endpoint. The New England Journal got the final report, and the director of the drug center, J. Richard Kraut, told the Cardiorenal Advisory Committee, Cardiorenal Division, which I was heading at the time, to get on with it and make a decision in 4 weeks, because this is about death. It's a big deal. So he gave us 4 weeks. We did it in 6. And the review led to non-approval, and interestingly enough, in 1980, the statistician on the thing, Gordon Pledger, and I published a paper explaining what the flaws of the trial were. How were we able to publish data on a drug we hadn't approved? You couldn't do that now. The reason is we never asked anybody. We just did it. I don't know if we could have gotten away with it. The trial itself had many nice features. It was double-blind. They were clever enough to hide the uric acid blood levels from the investigators. That was pretty clever, because that would have revealed who was on drug and who was not. So they hid it. That's pretty smart. They were randomized in blocks of 10. It was placebo-controlled. The entry criteria were Men and women aged 45 to 70. You had to have had a heart attack 25 to 35 days before you were entered. Documented by electrocardiogram, typical Hain history. And at least 2 of the following enzymes had to be increased, CPK, transaminase, and so on. They were not supposed to have cardiomegaly or overt heart failure, and they shouldn't be more than New York Heart Association Class 2 for heart failure, and they weren't supposed to have any life-limiting disease. And let me show you the reported overall results as presented. I got to be able to see them. So this was what the results in the trial were reported as. If you look at all deaths, there was a borderline finding, a p of 0.076, 62 to 44. Almost all the deaths were cardiac. Cardiac deaths were bordering on significant, but if you look at overall sudden deaths, it was nominally significant, 37 to 22. But interestingly enough, there was no trend on heart attack. Remember, it was the angioring reinfarction trial. The whole idea of the trial was that they were going to prevent recurrent heart attacks. There was nothing about heart attack deaths that even looked good. They then reported the data by cause and time. So if you look at all cardiac deaths for 0 to 6 months, there was a nearly 50% reduction, and it's now nominally significant. If you look at sudden deaths, that's where the action is. 24 to 6, p-value of 0.003. And in fact, the non-sudden deaths weren't even really leaning. So all of the action was on sudden deaths. That was not planned. They planned to use cardiovascular mortality originally, overall cardiovascular mortality. So sudden death was defined as death within 60 minutes of the sudden onset of symptoms or unobserved, unless the postmortem showed a heart attack. To be a heart attack, you had to have at least 2 elements of history, enzymes, or electrocardiographic. The trouble is, if you're not admitted to a hospital, what do you call someone who dies after 61 minutes of chest pain? Well, it's not sudden death because it's too late. It's not a heart attack because they don't have all those enzymes. They don't have all those other things. So it's not clear exactly how to do it. Part of the problem. The way the thing worked is the data were all sent to Sibagagi, who tried to catch early people who shouldn't have been entered, like they were too old or too early after the heart attack, and exclude them promptly. And maybe that would have been a good idea, maybe not. It could disrupt the randomization, but that's all pre-event. Nobody's had a heart attack or died of anything. And it would have been blinded. The impact would probably be small. We don't usually like that. They had an operating committee that was supposed to do an initial review of deaths and cause of death assignments. We don't actually, we never saw any records of what this committee did, but as I'll show you, at least briefly, when you look at the analysis of deaths, it's flagrantly biased. There was also a policy committee with significant people. They had other people doing audits. They had all of the appearances of being a spectacularly well-designed trial. Now, there were some built-in exclusions, not necessarily bad, they had something called the 7-day rule. If you weren't on treatment for 7 days, or if you were off treatment for 7 days, then events were excluded. The reason was the drug wouldn't act within 7 days, so you didn't want to do that, and the drug effect is gone after 7 days. So they wanted to exclude them. I don't think anybody would do that now, but it probably doesn't matter, and in fact, it doesn't matter here. If you died because of surgery unrelated to something else, they excluded you there. Interestingly, though, they also excluded people who were poorly compliant. That is, they found 3 patients who, after dying, had all the pills in their room, obviously hadn't taken them, and they excluded those. Now, that's not a large number of patients, but we would never allow that because there's a concern that compliance has something to do with outcome. There's some past experiences that might have said that. So that would be a funny exclusion. They did acknowledge in one of the papers that they had done that. In their early report, one patient was excluded for noncompliance, and they admitted it. They also excluded people who were, quote, ineligible. That is, people who mistakenly entered the trial because it was a day too late, or because they had evidence of heart failure, or whatever it is. And they dropped them in some cases after they died. If they had dropped them early, within the first few days having discovered it, you'd still worry about whether that would mess up the randomization or something, but it wouldn't have been so terrible. But these were people, 6 of them, who had died and then were excluded. Interestingly, they were all on Antarin. So you can see what the consequence of that is. Anyway, so the issues are, was the operations committee that calculated cause of death blinded? Who actually determined the cause of death? The cause of death assignment was very strange. We had some documentation. Every person who died had a new form that described the terminal event, filled out by the investigator. Interestingly enough, when the data were first submitted to us, the company didn't use the handwritten case report forms. They retyped it so that we could read it better, and submitted it to us that way. They were told by the person who was running the trial, reviewing the trial time, John Harder, that's no good, send us the handwritten ones. But in every case, they excluded the description of the terminal event. That form that the investigator filled out describing how the person died was omitted in 100% of the cases. When we got them later, then it came in. We had, but in general, it was very clear that the cause of death determination was not what the person who wrote the form had concluded. Somebody reached, that person might have said this is sudden death, but the final report would say, no, this was a heart attack. We had no idea who did that, where it came from. There was supposed to be a committee that did it, but we didn't know who did it. We don't know what the procedure for review of eligibility was. Why would eligibility be declared after death? Why? I mean, how come they only knew then? And we didn't know who did that. And then the late exclusion of poor compliers was sort of odd. Okay, so the results that were reported, as I showed you, was a near significant result in total cardiac deaths, highly significant reduction in early sudden deaths. It's not crazy to think the effect would be larger earlier. The risk after a heart attack is usually greater. The 6 months was arbitrary, not pre-specified, and so on. The whole outcome, sudden death, was not expected. Remember, platelet stickiness should have something to do with heart attacks, and it was called the Antarin reinfarction trial. There was discussion early about whether sudden death should even be included as one of the things. So anyway, ultimately, we didn't conclude any of these were supported because the cause of death analysis was biased, as I'll show you, and the late exclusions drove the favorable overall result, and as well as the 6-month result. So we had a suspicion about the cause of death assignment because the NIH told us that one patient had changed classification. How could that be? We didn't understand that. But we were naive to this whole thing. We had no idea that cause of death was so unstable. It had never come up. They never gave their analyses at trial run. What we did eventually, this wasn't adequate in retrospect, we looked at about 50% of the deaths using a description of the terminal event published on that form. As I told you, they weren't going to be submitted to us. And we looked at what we thought the terminal event was. We obviously should have done this in a blinded fashion, but we didn't. We were naive at the time. And we just found gross misapplication of the rules and inconsistency, generally tending to call a death on placebo sudden and a death on Antarin something else. That's what we found. I've got a bunch of examples, but I'm only going to show you 1 or 2. And these were all listed in our non-approval letter. And as I said, we published all this, which we would never be allowed to do now. So we said we looked at a 50% audit. We probably should have been blinded, blah, blah, blah. One of the things we found repeatedly was classification as sudden death despite evidence of a fresh occlusive thrombus or recent infarction at postmortem, which the protocol said meant it wasn't sudden death. It was supposed to be called an MI then. Although the NDA didn't explain how they did it. So one early placebo sudden death, postmortem examination showed old and recent right coronary artery thrombosis. A late placebo sudden death, postmortem examination showed recent right coronary artery thrombosis. A late anterine sudden death, there wasn't 100% bias in favor of against anterine, against placebo. Autopsy says specifically that the cause of death was acute thrombosis of right coronary artery, but was still called sudden death, which is weird. Another early placebo sudden death postmortem showed very recent extension of a posterior septal infarction. So there were lots of those, and I'm not going to read them all, but these slides will be made available, and you can read them all. But we found a lot of them. As I said, I don't have enough time to read them all. Okay, so the patients removed from the study as ineligible, but after death, favored anterine. So the data as originally presented was 62 to 43. It was nearly significant. And that excluded people who had poor compliance and so on. 6 of them, six patients. Poor compliance excluded 3, 1 on placebo and 2 on drug. And the ineligible, those numbers aren't in the right place, were 0 on placebo and 6 on anterine. They were felt to have not met the inclusion criteria, usually because it was a day late or whatever it is. And it's obvious that when there's 6 on the anterine group and 1 on and none on placebo, that's going to have a profound effect on the p-value. When you put them all in, the p-value, the nominal p-value is now 0.2. It's not anywhere close. It's interesting that the ones they decided were non-analyzable because they were not on it for 7 days or were off it for more than 7 days really didn't matter very much, and that's not too surprising. So the whole favorable result of the study overall is based on dropping patients who died after the study was over. The total of removing these people who were ineligible was 1 on placebo, 8 on anterine, and that gave you the whole finding. When you get to cause of death, cause-specific mortality, we know that these are very treacherous. We have a bias now toward looking at all cardiovascular mortality. It was clear that the 20 versus 6 in the 6 months was entirely due, as I've given these many examples, to calling deaths on anterine, calling deaths on placebo sudden death, and deaths on anterine something else, and that led to the 20.6. When you account for all of them and give them the finding, it goes completely away. So this was a very instructive thing for us. We just didn't understand how people could screw up a trial. Now everybody worries about who knew what, who saw the data, who was unblinded, and that's part of what we do when we go look, and it's absolutely critical because it's very easy to manipulate the results of a study. The end.